I want you to picture two 75-year-olds in your mind right now. Just, visualize them.
Okay, got him.
Right. So one of them is out there running a half marathon on a crisp Sunday morning. They're traveling across the globe without a second thought and easily keeping up with their teenage grandchildren.
A highly functional ager, yeah.
Exactly. But the other one is standing at the bottom of a short flight of stairs, just completely struggling to carry a single bag of groceries. They're dealing with this chronic, pervasive fatigue and managing a daily pill box that, it just seems to grow every single month.
Right, the classic manifestation of frailty.
Yeah, but here's the crazy part. If you pull out their birth certificates, they are identical. They have been on this planet for the exact same number of days. They've been breathing the same air, orbiting the sun the exact same number of times.
But biologically, they are miles apart.
Miles apart. They're living in entirely different physical realities. And today, for this deep dive, we actually have our hands on a stack of unyielding, hard DNA data that finally explains why.
It's the great paradox of human biology, isn't it? We track our lives by the calendar, marking off years and decades.
We just assume time moves at the same speed for everyone.
Right, we assume that. But your cells, well, they don't own a calendar. They keep their own time entirely. And what modern geroscience is revealing with just startling clarity is that the calendar is a deeply flawed, almost completely useless metric for understanding how old your body truly is.
Which brings us to our mission for today's deep dive. We are looking at a fascinating post hoc analysis published in the highly prestigious journal Nature Aging. It's looking at data from the Do Health trial.
And it's a monumental piece of research.
It really is.
Yeah.
And the best part is this isn't about some obscure $1 million gene therapy or, some speculative longevity biohack that you can't even pronounce.
No, we are talking about highly accessible interventions.
Right. We are looking at vitamin D, omega-3 supplements, and a basic home exercise routine. Stuff you probably have sitting in your kitchen cabinet or living room right now. Our goal is to decode this molecular data and understand if these everyday things can actually hit the rewind button on your cellular clocks.
It's a massive question to answer, honestly. Because, for decades, we have relied heavily on observational data.
Meaning we just sort of watch what people do and guess.
Basically, yeah.
Yeah.
We look at a population, we see the people who take certain supplements or stay active tend to live longer, healthier lives, and we just make an assumption.
But there are a lot of hidden variables there.
Oh, massive confounding variables.
Yeah.
The people who take omega-3s might also be the exact same people who eat more vegetables or get more sleep or avoid smoking. You just can't isolate the mechanism.
So it's just statistical noise.
Exactly. But what separates this Nature Aging paper from the noise is the rigorous nature of the Do Health trial itself.
Yeah.
This is a large-scale, randomized, double-blind, placebo-controlled clinical trial.
The gold standard.
The absolute gold standard. And we aren't just relying on self-reported questionnaires where they ask people, hey, how are you feeling today?
Right, because people lie or they just don't really know.
Yeah. Human recall is terrible. Instead, we are looking directly at the molecular level. The researchers are examining the DNA methylation patterns to see the individual and the additive effects of these interventions.
We are dissecting the fundamental why behind the cellular changes. But let's start with that fundamental why, because before we can even begin to understand how a simple home exercise program or like an omega-3 pill might change the aging process, we have to unpack how science actually measures aging on a cellular level.
Right. We need to define the ruler we're using.
Exactly. Because it's not as simple as looking at a skin cell under a microscope and deciding, well, it looks a little frayed around the edges. We are talking about something much, much deeper than that.
We are talking about the epigenome. The field of geroscience has undergone a complete revolution in the past decade, and it centers entirely around this specific concept.
So break that down for us. What is the difference between genetics and epigenetics?
Well, to understand epigenetics, you have to completely separate it from your underlying genetics. Your DNA, your genome, is your genetic instruction manual. Yeah. It is the hard drive you were born with.
And that doesn't change, right?
For the most part, no, it is fixed. It doesn't change whether you are 10 years old or 80 years old. But epigenetics, which literally translates to above genetics, refers to the chemical modifications layered on top of that fixed DNA.
Okay, so it's like the software running on the hard drive.
That's a great way to put it. These chemical layers dictate which specific genes are turned on, which are turned off, and critically, the volume at which they are expressed. And the primary modification we analyze in all these aging studies is called DNA methylation.
DNA methylation. So we are basically looking for physical chemical tags attached to the DNA strands.
Precisely. Methyl groups are these tiny chemical structures. They consist of just one carbon atom and 3 hydrogen atoms. And they attach themselves to specific sites on your DNA sequence.
I've heard them described like dimmer switches.
Yes. Think of them as billions of tiny molecular dimmer switches. When a methyl group attaches to a specific gene promoter, it generally turns the volume down on that gene. It suppresses its expression.
So it shuts the gene up.
Right. And when the methyl group is removed, the gene is turned back up. Now, what researchers discovered is that as we age, the pattern of these methyl groups across your entire genome changes in a highly predictable, systemic way.
Predictable how? Like, do we lose them or gain them?
It's a mix, actually. Some essential genes get inappropriately silenced because they gain methyl groups, while other highly detrimental genes suddenly lose their methyl groups and get turned on when they shouldn't be.
Oh, wow. So the whole system just kind of loses its mind.
Yeah, it becomes structurally chaotic. So epigenetic clocks are highly complex computer algorithms. They're trained to analyze hundreds of thousands of these specific methylation sites to quantify your true biological age based on that chaos.
Okay, let me see if I can try to visualize this for everyone listening. Chronological age is essentially the odometer on your car, right?
Sure, yeah.
It just rolls forward, counting the miles driven. It has absolutely no idea if those were smooth, easy highway miles or just brutal, pothole-filled, stop-and-go city miles. It just knows the absolute number.
The odometer is completely blind to context.
Right. But these epigenetic clocks are acting more like a master mechanic. They're opening up the hood, looking at the belts, checking the transmission fluid, measuring the brake pad degradation, and calculating the actual wear and tear on the machinery.
That is the perfect way to conceptualize it. The odometer tells you how far the car has traveled, but the mechanic tells you when the engine is actually going to fail.
And just like automotive diagnostics, the tools that researchers use to measure this have evolved really rapidly, haven't they?
Incredibly rapidly. In the Do Health BioAge study, the researchers didn't just rely on a single metric. They deployed multiple generations of epigenetic clocks to get a complete picture.
Which is crucial because looking at the study, they talk about first-generation clocks, second-generation, third-generation. It's kind of a lot to keep straight.
It is, but the distinction is vital. The first-generation clocks, things like the Horvath multi-tissue clock and the Hannum blood-based clock, were fundamentally limited.
How so? What were they doing wrong?
Well, it's not that they were wrong, it's how they were built. They were constructed by taking thousands of DNA samples and training a machine learning algorithm to predict the chronological age of the person the sample came from.
Wait, hold on. If the algorithm is just trained to guess how many birthdays you've had, doesn't that defeat the entire purpose?
It really does.
I mean, if I want to know how fast my engine is decaying, I don't need a sophisticated blood test to just confirm what my driver's license already says.
You've hit on the exact limitation that drove the entire field to pivot. The first-generation clocks are incredibly accurate at predicting chronological age. I mean, they are mathematically brilliant.
But they miss the point of biological aging.
Exactly. Because they were mathematically anchored to the calendar, they often failed to capture the nuances of health span. They couldn't consistently differentiate between a 75-year-old marathon runner and a 75-year-old who is bedridden.
Because to the algorithm, they're both just 75.
Right. And this realization gave birth to the second-generation epigenetic clocks, which are the true heavyweights in this Nature Aging paper. We were talking about algorithms like PhenoAge, GrimAge, and the recently updated GrimAge 2.
So how did they train the algorithms differently this time? If they aren't using the calendar as the anchor, what are they measuring the DNA against?
They anchored the algorithms to physical decay and death.
Oh, wow. That is a dark pivot.
It is dark, but it's highly functional. Instead of training the computer to predict chronological age, the developers trained these second-generation algorithms on massive data sets of clinical data reflecting physiological deterioration and mortality risk.
Can you give an example of how they actually did that?
Let's look at PhenoAge, for example. To build PhenoAge, researchers first used data from thousands of individuals who were tracked over 23 years.
Okay, a huge longitudinal study.
Right. They identified 10 specific clinical blood markers, things like glucose levels, white blood cell counts, and markers of kidney function. These markers strongly predicted whether someone was going to develop a disease or die.
So they created a risk profile based on standard blood work.
Yes, they combined those markers into a single phenotypic age score. And only then did they look at the DNA methylation data, training the algorithm to predict that specific health-based score, not the calendar age.
Oh, I see. That completely flips the paradigm. You aren't measuring time anymore. You are measuring the physical manifestation of risk.
Yes, and Grimmage took this even further.
Grimmage is such an intimidating name for a scientific tool.
It really earns the name, though. Grimmage was developed by identifying the DNA methylation patterns that perfectly surrogate specific plasma proteins floating in your blood.
Which protein?
Proteins that are heavily associated with cardiovascular disease, systemic inflammation, and all-cause mortality.
Yeah.
Plus, they combined it with a surrogate marker for how much you've smoked in your lifetime.
So it's basically a molecular Grim Reaper.
Pretty much.
Yeah.
When Grimmage or phenoage indicates that your biological age is higher than your chronological age, it is a glaring siren. It strongly predicts your immediate risk of age-related morbidity.
Okay, so the second generation is predicting their trajectory toward disease. But as I was reading through the methodology, there is a third-generation clock they use called Dunedin pace.
Ah, yes. Dunedin pace.
And it seems to operate completely differently from the rest. The Do Health researchers seem to lean heavily on it in the paper.
Dunedin pace is just a master class in longitudinal study design. The other clocks we discussed, they measure your absolute biological age.
Like giving you a static number.
Right. They give you a static number, estimating how biologically old you are at the specific moment the blood is drawn. But Dunedin pace doesn't measure age, it measures velocity.
Velocity, like how fast you are aging right now.
Exactly. It stands for pace of aging, calculated from the epigenome. It was developed using a very specific group of people, the Dunedin study cohort in New Zealand.
What makes that cohort so special?
It's a group of roughly 1,000 individuals who are all born in the exact same year, in the exact same town. And they have been tracked extensively, getting poked and prodded for their entire lives.
Oh, that's brilliant. Because they share the exact same chronological age, any biological difference between them is purely a difference in how fast their bodies are decaying.
Precisely. By analyzing how their organ systems, their lungs, their kidneys, their cardiovascular systems, how they declined over time, researchers built an algorithm that measures your pace of aging.
So it spits out a rate, not an age.
Yes. It tells you how many biological years you are aging for every single calendar year that passes.
To return to our car analogy, if age is the mechanic assessing the current overall condition of the engine block,
then Dunedin pace is the speedometer on the dashboard. It tells you exactly how fast the damage is accumulating right at this very second.
Okay, hold on. I need to wrap my head around this for a second. We have Horvath and Hannum predicting the calendar. We have phenoage looking at systemic physiological decline.
Right.
We have Grimmage looking at cardiovascular and mortality-linked proteins. And we have Dunedin pace measuring the speedometer.
Correct.
If all of these things are fundamentally measuring the aging process, aren't they highly redundant? I mean, if an intervention like vitamin D truly works, shouldn't it just turn all of these clocks back simultaneously? Why go through the exhaustive process of tracking every single one of them?
It is an incredibly important point to clarify, because the lack of redundancy is actually the key to understanding human longevity.
What do you mean? Aging isn't just one thing.
No, not at all. Aging is not a single centralized switch that slowly gets turned off. It is a highly complex, decentralized, systemic failure occurring across multiple independent pathways.
So different parts of the car break down at different times.
Exactly. Your immune system decays at a different rate and through entirely different mechanisms than your metabolic system. And that ages differently than your brain. These different epigenetic clocks are capturing different fragments of that systemic failure.
Okay, that makes sense. So what is Grimmage specifically capturing?
Grimmage is heavily weighted toward immune system dysfunction and metabolic dysregulation, whereas phenoage captures a broader, more generalized multi-system decline.
So by utilizing a diverse array of these validated clocks, researchers can see exactly which biological levers an intervention is actually pulling.
Exactly. If a supplement changes Dunedin pace but doesn't touch Grimmage at all, it tells you something highly specific about what that molecule is doing inside the body.
It maps the mechanism of action.
Yes. We have a highly sensitive suite of diagnostic tools ready to measure the exact nature of the wear and tear.
So if we have the ruler, what are we measuring? Because you can't just hand someone a random bottle of pills, tell them to do some jumping jacks, and hope for the best.
Definitely not.
To prove that these specific interventions actually change the epigenetic source code, you need a highly controlled environment. Which brings us directly to the anatomy of the Do Health trial itself.
The population they selected for this specific epigenetic sub-study is really critical to contextualizing the results.
Yeah.
The overall Do Health trial spanned multiple European countries.
But they didn't use everyone for this specific DNA analysis, did they?
No. For this specific DNA methylation analysis, they isolated 777 participants exclusively from the Swiss cohort.
Just the Swiss. Okay.
Yeah, and the demographics are important here. The average age at baseline was 75.5 years, and roughly 60% of the cohort were women. But the most vital piece of context is the health status of these individuals.
Right. I noticed that in the supplementary data, they weren't sick.
Not at all. This was an exceptionally healthy population.
Yeah, over 53% of them met the incredibly strict nurses' health study criteria for being healthy agers.
Which is a very high bar to clear.
It really is. That means they were totally free of major chronic diseases. No history of cancer, no cardiovascular disease, no diabetes. They had no major physical disabilities, and their cognitive function was completely intact.
And on top of that, an astonishing 88% of them were physically active.
Right. With nearly 60% engaging in physical activity more than 3 times a week, I think the paper said only 12% of the entire cohort was considered completely inactive.
So we're talking about 75-year-olds who are actively participating in life. They are living independently, moving their bodies, and successfully avoiding catastrophic disease.
Which is great for them, but doesn't that make the trial much harder for the researchers?
Exponentially more difficult.
Mm.
In medical research, it is almost always easier to demonstrate the efficacy of an intervention when you test it on people who are deeply sick or severely deficient.
Like, if I have terrible blood pressure, any pill you give me is going to look like a miracle.
Exactly. If you take a patient with dangerously high cholesterol and give them a statin, you will see a dramatic, immediate drop. The physiological ceiling is so low that any intervention looks highly effective.
But trying to further optimize the biological age of a 75-year-old who is already a highly active, healthy ager.
That requires an intervention that is fundamentally altering the baseline mechanics of cellular health. It is the ultimate test of true optimization versus basic remediation.
So, to figure out if they could clear that high bar, the researchers deployed what is called a 2x2x2 factorial design over a 3-year intervention period.
A very robust statistical model.
Let's walk through the actual interventions because, as we said, this isn't science fiction. These are highly accessible tools.
The first intervention was 2,000 international units of vitamin D3 taken daily.
A pretty standard moderate dose.
Very standard. The second was 1 gram per day of omega-3 fatty acids, but it is vital to note the source here.
Yeah, let's talk about that because this was not your standard off-the-shelf fish oil from the local pharmacy.
No, it was specifically 330 milligrams of EPA and 660 milligrams of DHA derived entirely from marine algae.
Why marine algae instead of fish oil? Fish oil is literally what everyone has in their medicine cabinet right now.
It comes down to standardization, purity, and sustainability. Most people don't realize this, but fish do not actually produce omega-3 fatty acids themselves.
Wait, really?
No, they accumulate them by eating microalgae. By going straight to the source and utilizing marine algae, the researchers ensure a highly purified, consistent ratio of DHA to EPA.
And I imagine it's like cleaner.
Much cleaner.
Yeah.
You completely avoid the risk of heavy metal contamination or lipid oxidation that often plagues low-quality commercial fish oil. You get the pure active compound without the toxic baggage.
Oh, that makes a lot of sense. And what was the third intervention?
The third was a simple home exercise program, referred to as SHE in the literature. It consisted of a 30-minute strength training routine performed 3 times a week.
But they had a control group for the exercise too, right?
Yes, and this is where the trial design really shines. To ensure a rigorous control, the participants who are not assigned to the strength training group didn't just sit on the couch.
That would skew the data because just doing something makes people feel better.
Exactly, the placebo effect of participation. So the control group was assigned a sham exercise routine consisting of basic joint flexibility movements. It controlled for the psychological and minor physiological effects of simply participating in a health routine.
This 2x2x2 factorial design is really brilliant from a statistical standpoint because by randomizing participants into 8 different treatment arms, they aren't just isolating one variable.
Right, they get to see the whole picture.
They can evaluate the main effect of each intervention individually. Like they can analyze the DNA of everyone who took vitamin D against everyone who took a placebo, but they can also look at the interactive additive effects.
Which is how life actually works.
Yeah. They can see what happens with vitamin D alone, omega-3 alone, exercise alone, or vitamin D stacked with omega-3, or omega-3 stacked with exercise, or all three combined.
It perfectly mirrors the complexity of human behavior. People rarely do just one thing to improve their health. They layer interventions.
But from a public health perspective, we have to ask, why did the researchers choose these three specific interventions out of thousands of possible longevity compounds?
It's a fair question.
I mean, why not metformin? Why not rapamycin or any of the exotic, incredibly expensive peptides dominating the longevity conversation on social media right now?
It fundamentally comes down to scalability, safety, and a crucial biological concept known as pleiotropy.
Pleiotropy, break that down for us.
Pleiotropy occurs when a single intervention has diverse, simultaneous protective effects across multiple independent organ systems.
Like a master key.
Exactly. Vitamin D, omega-3s, and basic strength training are highly pleiotropic. They are known to independently modulate systemic inflammation, support immune cell function, maintain muscle and bone integrity, and improve metabolic lipid profiles.
They hit a lot of targets at once.
And critically, they are inexpensive and widely accessible to the global population. If your goal is to design a public health intervention to slow the burden of aging on a societal level, you need interventions that could be deployed to 1 billion people safely.
Right. You can't just hand out experimental drugs to everyone.
No, you cannot mass prescribe experimental immunosuppressive pharmaceuticals without catastrophic risk. But you can absolutely safely encourage a standardized algae supplement and 30 minutes of bodyweight squats in the living room.
That makes total sense. So we have our 777 healthy Swiss participants. We have our 3 accessible interventions mapped out in this exhaustive factorial design. And we have the most advanced epigenetic clocks in the world ready to measure the wear and tear over a 3-year period.
The stage is set.
The stage is set. So when the 3 years were up, they unblinded the data. They looked at the single interventions first, asking the data, did vitamin D work on its own? Did exercise work on its own? Did omega-3 work on its own?
And the results were very surprising.
Yeah, when the results came in, there was an undeniable MVP, a solo star that completely outshone the others.
The data was definitive. When evaluating the single interventions in isolation, daily omega-3 supplementation was the standout performer.
Just the omega-3?
Just the omega-3. Vitamin D supplementation alone, despite its vast pleiotropic benefits, did not significantly alter any of the epigenetic clocks on its own.
Wow. And the exercise.
The simple home strength training program alone also did not induce a significant change in the clocks. But the omega-3 alone significantly reduced the biological age and the pace of aging across 3 of the 4 advanced 2nd and 3rd generation clocks.
Let's look closely at that. Omega-3 alone slowed down pheno-age, Grimmage-2, and Dindon pace, but it did not significantly move the 1st generation Horvath or Hannum clocks.
Which brilliantly validates exactly what we discussed earlier.
Right. The 1st generation clocks just track the calendar, but the advanced clocks track true physical decay. So the omega-3 was actively slowing down the rate of biological deterioration.
That's the power of targeting the right mechanism.
But we need to translate the math into reality. The study relies on standardized units, which makes the graphs look really clean and impressive to scientists, but it doesn't mean much to the average person.
Standard deviations don't translate well to everyday life.
No, they don't. When they converted those standardized effect sizes into real-world time, how much younger were these cells?
The omega-3 supplementation over the course of the 3-year trial slowed the biological clocks by the equivalent of 2.9 to 3.8 months, depending on the specific clock being measured.
Okay, I am going to be completely honest here. When I first read that figure in the supplementary tables,
We were impressed.
my initial reaction was incredibly underwhelming. I mean, you take a pill every single day for over 1,000 days, and your reward is a 3-month rollback. That does not sound like the fountain of youth. It sounds like a rounding error.
That is a very common and completely understandable human reaction. We are so conditioned to look for immediate, dramatic transformations in medicine.
We want the overnight fix.
Exactly. But to understand the true impact of this data, you have to shift your perspective from individual short-term clinical urgency to the vast scale of population-level geroscience.
Okay, zoom out for us.
3 months might seem trivial to one individual over a 3-year snapshot. However, that compounding effect culminates in years of additional highly functional health span. Furthermore, consider the staggering economic and societal implications.
What happens when millions of people do this?
Delaying the onset of devastating age-related conditions like cardiovascular disease, dementia, or severe physical frailty by even an average of 3 months across an entire national population creates massive economic returns.
Because they aren't in hospitals.
Right. It significantly reduces the burden on acute healthcare systems, frees up billions in medical spending, and dramatically improves the collective quality of life. It is not about living forever. It is about extending the period of life spent completely free from debilitating disease.
A small shift in the trajectory creates a vastly different destination.
Beautifully said.
It's like if a rocket is launching to Mars, a 1-degree shift in trajectory in the first minute of flight dictates whether it lands perfectly on the planet or misses it by a million miles. The omega-3 is shifting the trajectory.
That's exactly what the data shows.
But as impressive as the omega-3 was on its own, the real magic of this deep dive emerges when we look at the additive effects. Because people in the real world don't just do one thing. They stack their habits.
This is where the 2x2x2 factorial design truly pays dividends. The researchers found compelling evidence of synergistic additive treatment effects, specifically when looking at the pheno-age clock.
So the combinations worked better.
Much better. As we noted, vitamin D and exercise didn't move the needle much in isolation. However, when you combine them with the omega-3, the biological age deceleration became significantly more pronounced.
They amplify each other.
Yes, the combination of omega-3 with vitamin D, or omega-3 with exercise, or the ultimate combination of all three interventions stacked together yielded standardized effect sizes that were demonstrably stronger than the omega-3 alone.
Let's use a biological metaphor here, because I think a lot of people assume that if you take 3 supplements, you just get 3 independent benefits running on parallel tracks.
Like they don't interact at all.
Right. But the body doesn't work like that. It's more like a complex security system. Think of vitamin D as the specific key required to unlock the cellular door.
I like this analogy.
If your vitamin D levels are too low, the door remains locked. You can flood the bloodstream with the highest quality omega-3 in the world, but it just bounces off the cell wall because it literally can't get inside.
It lacks access.
Exactly. But when you introduce the vitamin D, you unlock the receptor pathways. Then the exercise acts as the structural reinforcement. It primes the tissue and increases blood flow.
It prepares the environment.
Right. And then the omega-3 enters the cell and acts as the specialized mechanic, physically repairing the inflammatory damage and altering the epigenetic expression. They are mutually dependent.
That is an excellent, highly mechanistic way to view it. And the researchers didn't just observe this high-level synergy and stop there.
They dug deeper.
They drilled all the way down into the specific DNA methylation surrogate markers to prove exactly how that mechanic was fixing the engine. We really need to look closely at the Grimmage clock components.
Let's do it.
Remember, Grimmage is built upon epigenetic estimates of specific plasma proteins floating in your blood. Higher levels of these specific proteins strongly correlate with a higher risk of death and metabolic collapse.
This was honestly the most illuminating part of the entire paper for me, because it strips away the abstraction of, biological age and ties it directly to tangible, functional proteins acting inside the vascular system.
It grounds the math and biology.
Exactly. When they looked at the omega-3 intervention alone, it successfully modified 3 of these detrimental proteins, PAI1, leptin, and TIMP1.
A solid result.
But when they stacked all 3 treatments together, the omega-3, the vitamin D, and the exercise, they achieved significant synergistic downregulation in 4 critical proteins, PAI1, TIMP1, B2M, and GDF15.
Which is a massive shift in the protein profile.
But we have to break down what these proteins actually do, otherwise it just sounds like alphabet soup.
Let's start with PAI1 because its downregulation was highly significant. PAI1 stands for Plasminogen Activator Inhibitor 1.
Okay, what is its day job?
Its primary role in the body is the regulation of blood clotting. Specifically, it inhibits the enzymes that naturally break down clots. As human beings age, our baseline levels of PAI1 steadily rise.
Which means we can't break down clots as well.
Exactly. This creates a hypercoagulable state, meaning your blood is far more likely to form dangerous clots, leading to a drastically increased risk of thrombosis, strokes, and heart attacks.
And beyond just clotting, isn't PAI1 heavily implicated in cellular senescence?
Yes, intimately so. Senescent cells are often referred to in the literature as zombie cells.
Because they refuse to die.
Right. They are damaged cells that refuse to die, but they stop dividing. Instead, they just sit in your tissue and secrete a highly toxic soup of inflammatory molecules that accelerate the aging of all the healthy cells around them.
And PAI-1 is part of that soup.
PAI-1 is a major component of that toxic soup. The fact that the combined Do Health interventions significantly reduce the epigenetic signature of PAI-1 means they are actively dismantling a primary driver of cardiovascular risk and systemic inflammation.
That alone is huge. But let's move to GDF-15, growth differentiation factor 15. This one responded beautifully to the combination of all three treatments, and its biological role is fascinating.
GDF-15 is widely considered a primary biomarker of mitochondrial dysfunction and deep cellular stress.
Mitochondria being the powerhouse of the cell, of course.
Exactly. They are the microscopic power plants operating inside your cells, generating the energy required for survival. As we age, these power plants begin to fail. They leak, they become inefficient, and they generate massive amounts of oxidative stress.
And the cell notices this.
When a cell senses this mitochondrial failure, it panics, and it produces huge amounts of GDF-15 as a distress signal.
So it's an alarm bell.
A very loud alarm bell. High levels of GDF-15 are heavily correlated with physical frailty, muscle wasting, and all-cause mortality.
So by utilizing the additive combination of omega-3, vitamin D, and exercise to suppress the epigenetic surrogate of GDF-15.
The intervention is likely signaling a fundamental improvement in mitochondrial efficiency and a drastic reduction in cellular panic.
That leaves us with B2M and TIMP1. The literature notes that B2M, or beta-2 microglobulin, is a classic marker of severe inflammatory and infectious conditions.
Right, another marker of systemic stress.
And TIMP1 requires a bit of translation. It stands for tissue inhibitor of metalloproteinases.
It's a mouthful. To understand TIMP1, you have to understand matrix metalloproteinases. Think of matrix metalloproteinases as microscopic, highly specialized scissors that travel through your body, cutting up the extracellular matrix.
The matrix being the structural scaffolding that holds your cells together.
Exactly. This cutting is totally necessary for normal tissue repair and remodeling. You have to break things down to rebuild them. TIMP1 is the protein that stops the scissors. It prevents them from cutting too much.
Keeping the balance.
But as we age, the balance is lost. TIMP1 becomes highly dysregulated.
And when TIMP1 gets dysregulated, the microscopic scissors just go rogue. They start cutting up the structural scaffolding inappropriately, which degrades the tissue.
And that has terrible consequences.
Right, because critically, it creates a chaotic, broken environment where cancer cells can easily detach, migrate, and spread.
You nailed it. Dysregulated TIMP1 is heavily linked to tumor progression and cancer metastasis. And this brings us to the most profound synthesis of the entire deep dive.
Pull it all together for us.
When you see these specific, highly dangerous proteins, PAI-1 driving blood clots, GDF-15 signaling failing mitochondria, B2M driving severe inflammation, and TIMP1 creating an environment primed for cancer.
All going down.
Yes. Seeing them all being epigenetically downregulated by these simple interventions creates a perfect bridge to the physical reality of the patients in the trial.
Right, because in earlier publications stemming from the broader Do Health trial, which tracked over 2,100 participants across Europe, the clinical outcomes were staggering.
The clinical data was incredible.
The researchers reported that the combination of all three interventions led to a 39% reduction in the risk of becoming physically pre-frail.
39%.
And even more incredibly, the combined intervention resulted in a 61% reduction in the incidence of invasive cancer over the 3 years.
For a long time, we just had those clinical numbers. We knew the patients were avoiding cancer and maintaining their physical strength, but we couldn't see the gears turning underneath.
We didn't know how they were doing it.
Exactly. Now, with this nature-aging epigenetic data, the molecular mechanism is laid completely bare. The downregulation of PAI-1, B2M, TIMP1, and GDF-15 provides the exact cellular explanation for why these 75-year-olds remained robust.
They were changing the terrain.
The interventions weren't just masking symptoms. They were fundamentally repairing the structural integrity of the cellular environment, making it deeply inhospitable to frailty and cancer.
It is the ultimate scientific full circle moment. The DNA methylation changes dictate the protein expression dictates the cellular environment, and the cellular environment dictates whether you spend your 75th year running a marathon or struggling with groceries.
It's all connected.
But this brings us to a massive nuance in the data, one that modern medicine is finally starting to take really seriously. Biological individuality.
Oh, this is crucial.
Because we know the combination works, but did it work equally well for every single person in the trial?
It did not, and the researchers deliberately designed the analysis to expose these exact variations. This is the absolute cornerstone of personalized medicine. We can no longer look at human beings as identical biological machines.
So they broke the data down by subgroups.
Yes. The researchers conducted rigorous subgroup analyses to determine if baseline characteristics, things like sex, BMI, and crucially, baseline nutritional status, altered the efficacy of the interventions.
And the variations were significant. For instance, the beneficial effect of omega-3 on the pheno-age clock was demonstrably larger for individuals who started the trial with adequate levels of vitamin D.
Specifically, blood levels at or above 20 nanograms per milliliter.
Which perfectly reinforces the locking key metaphor we used earlier. If you are chronically deficient in vitamin D, the omega-3 simply cannot perform its epigenetic repair to its full potential. The body requires a foundational baseline of certain nutrients before it can leverage others.
They also observed that the additive effects of combining the treatments were more robust in women than in men.
Why is that? Do we know?
The mechanisms behind that sex-specific difference require further investigation, but it's a very clear signal in the data. But honestly, the most critical personalized finding in the entire paper revolved around the baseline blood levels of the omega-3 fatty acids themselves.
How much they had before the trial started.
Exactly. Before a single pill was ever taken, the researchers measured the circulating levels of EPA and DHA in the participants' blood.
And what did they find?
They discovered that the epigenetic age deceleration was significantly stronger in individuals who began the trial with lower baseline levels of these fatty acids, specifically below 100 nanograms per milliliter.
And there is a really vital piece of geographical context here. This specific epigenetic analysis was conducted entirely on the Swiss cohort of the Do Health trial.
Switzerland is a landlocked country.
Right. Their traditional diet is not heavily reliant on fresh, cold water oceanic fish. The researchers highlighted that this Swiss subgroup actually presented with notably lower baseline omega-3 levels compared to the broader Do Health population drawn from other, more coastal European countries.
Geography dictated baseline biology. Because this specific Swiss cohort was starting from a state of subclinical deficit, introducing a highly purified 1-gram daily dose of marine algae, omega-3 filled a massive nutritional void.
The body was practically begging for it.
The biological system was completely starved for these specific lipids, so when they were finally introduced, the epigenetic response was profound and immediate.
So let's think about this practically. If you were listening to this right now and you live on the coast and your diet consists of wild-caught salmon and mackerel 3 times a week, your baseline omega-3 levels are likely already highly saturated.
Very likely, yes.
If you go out and buy a premium algae supplement based on this study, is it essentially just an expensive way to create highly enriched urine?
Well, based on the mechanisms revealed in this data, that is a highly logical conclusion.
So you wouldn't see the age reversal?
Probably not. Your biological sponge is already completely full. Adding more water to a saturated sponge doesn't change the nature of the sponge, it just runs off. This is the danger of blindly adopting longevity protocols without understanding your own biochemical state.
You have to know where you're starting from.
Exactly. The interventions in this trial exerted their most powerful epigenetic effects when they were correcting a deficit. More is rarely better in biology. Optimization is about achieving homeostasis.
So if you already have enough, the pill doesn't do much.
If your diet is already providing ample EPA and DHA, an additional gram via supplementation is unlikely to yield these same dramatic decelerations in epigenetic aging. Knowledge is only powerful when it is understood and applied to your specific individual context.
That is the most important takeaway of the entire discussion. You have to measure your baseline before you try to change it.
Measure twice, cut once.
Exactly. Now, as incredible as this data is, no study is without its flaws.
Yeah.
If we are going to look at this objectively, we have to acknowledge the boundaries and limitations of the research. Where does this data fall short?
Critical appraisal is essential. And to their credit, the researchers are highly transparent about their limitations. First and foremost is the duration of the trial.
3 years.
3 years is a significant commitment for a randomized clinical trial, absolutely. But in the context of human aging, it is merely a snapshot.
We don't know what happens in year 10.
Right. We do not know if these epigenetic changes are permanently locked in, if they eventually plateau, or if the benefits continue to compound logarithmically over 10 or 20 years. Secondly, They were only able to measure the DNA methylation data at 2 specific time points.
The baseline measurement on day 1 and the final follow-up at year 3. Wait, why is that a problem? You take a picture before the intervention and you take a picture after. Isn't that enough to show a difference?
It is sufficient to show a difference, but statistically, it is highly imperfect. Relying on only 2 time points significantly increases the potential for measurement error.
How so?
Imagine trying to chart the trajectory of a stock price by only looking at its value on January 1st and December 31st. You completely miss all the volatility and nuance in between.
Ah, okay, that makes sense.
However, in the realm of statistical modeling, an increase in measurement error typically biases the effect estimates toward the null.
Meaning the static makes it harder to hear the underlying signal?
Precisely. It makes it mathematically more difficult to prove a statistically significant result.
Oh, so the fact that they found anything at all.
Exactly. The fact that the researchers still observed robust, significant decelerations in biological age across multiple highly sensitive clocks, despite the increased measurement error of only having 2 time points, implies that these findings are actually highly conservative.
The real effect could be way bigger.
The true underlying biological effect of the omega-3 and the combined interventions might be considerably stronger than what the standardized data indicates.
That is wild. Another boundary of the data is the population itself. We spent time detailing how incredibly healthy these 75-year-old Swiss participants were.
The healthy agers.
Right. But that inherently means we cannot confidently extrapolate these results to a population that is already deeply frail, severely sedentary, or battling advanced chronic disease.
No, we really can't.
If someone's cellular environment is already completely overwhelmed by massive systemic inflammation and advanced senescence, handing them a 1-gram omega-3 pill and a light exercise routine might be like trying to put out a forest fire with a garden hose.
That is a very accurate assessment of the limitations.
Well,
Contextualizing these results against other major geroscience trials really helps illustrate this complexity. Consider the calorie trial, which is one of the most famous trials in the longevity space.
right, the calorie restriction trial.
Yes. Calorie tested the effects of strict long-term caloric restriction on human biological aging. When they applied the Dunedin pace clock to the calorie participants, they found that caloric restriction induced a 2 to 3% reduction in the pace of aging.
And in the Do Health trial we've been discussing, the omega-3 intervention reduced the Dunedin pace clock by roughly 1%. So caloric restriction is significantly more powerful at slowing down the immediate speedometer.
On that specific clock, yes. But here is where the complexity of aging reveals itself. In the calorie trial, while caloric restriction dramatically slowed the Dunedin pace speedometer, it did not significantly reverse the phenoage or Grimmage clocks.
Wait, really? It didn't touch them?
It didn't meaningfully alter those specific protein surrogates predicting mortality. Conversely, in the Dune Health trial, while the effect on Dunedin pace was more modest, the interventions had profoundly strong effects on phenoage and Grimmage too, rolling them back by nearly 3 to 4 months.
That is incredible. It proves that there is no single monolithic pathway to stopping aging.
Aging is a hydra.
Caloric restriction is pulling a massive metabolic lever that slows down the absolute rate of systemic decline. But omega-3, vitamin D, and exercise are pulling entirely different levers.
Much more specific levers.
They're acting as targeted mechanics, actively repairing the specific inflammatory damage and altering the precise proteins, like PAI1 and TIMP1, that dictate physical frailty and disease risk. It takes an orchestra of different interventions to truly alter the biology of aging.
It is a biological symphony, and this Nature Aging paper provides definitive randomized clinical proof that if we apply the right combination of nutrients and physical stimulus, we can literally change the music being played by our DNA.
So let's tie this all together. We have journeyed through the microscopic architecture of human longevity today. The ultimate takeaway is one of profound agency.
Yes, agency is the key word.
Biological age is not a fixed, inescapable sentence handed down by the calendar. You are not just a passive passenger strapped into a slowly deteriorating machine. You have access to the steering wheel.
You have control over the inputs.
This Do Health data proves that omega-3, specifically high-quality marine algae DHA and EPA, is a potent, independent intervention capable of slowing the epigenetic decay of your cells, especially if you are currently deficient.
Correct.
But more importantly, true biological optimization requires synergy. By combining that omega-3 with adequate baseline vitamin D and the mechanical stimulus of a simple home-strength routine, you create an additive multi-system cellular repair sequence.
You build a stronger environment.
You literally downregulate the molecular proteins responsible for blood clots, failing mitochondria, and cancer progression. The power to alter your trajectory is sitting right there in your daily habits.
It is a remarkable synthesis of how daily lifestyle choices physically intertwine with our genetic expression. But before we conclude, I want to leave you with one final, deeply provocative detail that was quietly buried in the methodological protocols of this research.
Oh, I love buried details. Lay it on us.
When the Do Health researchers were establishing the parameters for drawing the blood samples used for this DNA methylation analysis, they mandated that all blood must be drawn strictly in the morning and strictly in a fasting state.
Just to keep things consistent.
Well, yes, but why do they have to control the timing so ruthlessly? Because cutting-edge research has recently revealed that epigenetic age is not static. It actually oscillates wildly throughout the day.
Wait, hold on. It fluctuates based on the time of day?
Yes. Your biological age is entirely fluid across a 24-hour period. According to recent circadian biology studies, your epigenetic clocks are biologically older at solar noon than they are at midnight.
Are you serious?
Entirely. Your DNA methylation patterns literally ebb and flow in perfect synchrony with your circadian rhythms, your cortisol spikes, and your daily metabolic demands. At midday, when your body is digesting food, dealing with physical stress, and fighting gravity, your cells are literally older.
And at night?
At night, during deep sleep, the epigenetic clocks roll backward as the system repairs itself.
That is absolutely mind-bending. You are aging and rejuvenating every single day on a microscopic scale. Think about the profound implications of that for the future of human health.
It changes everything we know about tracking health.
Imagine a world where, a few years from now, instead of glancing at a smartwatch to see if you hit your arbitrary 10,000 steps, you are checking a continuous, real-time epigenetic monitor.
A literal biological clock on your wrist.
A device that tells you exactly how fast your DNA is aging at that exact second. Will we soon be modifying what we choose to eat for lunch? Or deciding whether we desperately need a 20-minute afternoon nap based entirely on real-time feedback from our cellular clocks?
It's entirely possible.
Imagine how having that immediate, undeniable view into your own microscopic decay might completely alter the way humanity chooses to live. It changes the entire paradigm. It's no longer just about optimizing the decades of your life. It is about optimizing the specific hours in your day, something for you to deeply ponder until next time.